Assalamu alaikum students and welcome to the lecture 21 of the cell signaling course. Today we will talk about a very important cellular process called apoptosis. This is the overview of my talk. I'll briefly introduce apoptosis to you, and then I will talk about different components of apoptosis such as caspases and BCL2 family of proteins, and then we'll talk about two very important apoptotic pathways. So apoptosis is a self-destruction in an orderly and highly controlled manner. This is the coordinated demolition of cellular structure. So coordinated demolition of cellular structures by special proteases called caspases. Now, an apoptotic cell has characteristic changes in its morphology, and these characteristic changes are condensation of chromosomes, it is degradation of DNA, into fragments, it is cell shrinkage, fragmentation of the nucleus, and disassembly of the membrane into something that is called apoptotic bodies. So apoptosis, as I mentioned, is a programmed cell death. So it is actually involved in very important biological processes. Apoptosis plays a very important role during differentiation and development. It plays a very important role in elimination of cells during differentiation of and development, elimination of cells during immune response. It plays important role in tissue homeostasis. Plays an important role in elimination of tumor cells. So, in a normal cell, in a normal body, around 10 billion cells, they die through apoptosis each day. Especially in our immune cells, many cells, they actually die due to apoptosis. So apoptosis is actually strictly controlled. And, it is strictly controlled by signals that can be from within the cells. These signals can be DNA damage, for example, excessive signaling, or these signals can be from outside the cell. For example, fast ligand or TNF we studied the other day. So they can activate what is called death receptors. So these are the receptors that have death domain, and we studied about that. Similarly, many signaling pathway, actually all of the signaling pathway that form, stress that responses or DNA damage responses, they have some sort of link with apoptosis. So, apoptosis, as I mentioned, is self-destruction in an orderly and highly controlled way. It's coordinated demolition of intracellular structures by the member of caspase family of proteases, and the correct, it causes characteristic changes in the morphology, as I described. Condensation of the chromosomes, fragmentation of the DNA, cell shrinkage, fragmentation of the cell nucleus, or apoptotic body formation due to disassembly of the membranes. Now, this is an indispensable part of the development and function of the organism. So, uh, it is required for tissue homeostasis. It is required for, during development. It's required during immune response. It is required to avoid tumors. Many of the tumors have mutation in apoptotic machinery, so they can't die because of the programmed cell death. Now, there are two major apoptotic pathway. One is called intrinsic pathway. So this gets signals from inside the cell. It is also called mitochondrial pathway. And the other one is called extrinsic pathway. So this is death receptor pathway. We'll talk about them in detail later on. So there is extrinsic pathway, and there is intrinsic pathway. So you have intrinsic pathway, you have extrinsic pathway. So extrinsic pathway involves, Fas ligand. It involves TN. It can be activated by TNF cytokine. The intrinsic pathway can be activated by stress, DNA damage. It can be activated by excessive signaling. And your extrinsic pathway binds to Fas, which is CD95, I believe. Let me check. Yes, and then it can also bind to TNF receptor. And then the intrinsic pathway activate proteins like BAX. For example, BAD we studied, or BAX, which are, which activate mitochondrial apoptosis. So this is mitochondrial apoptosis. That's why intrinsic pathway is also called, mitochondrial pathway. So your extrinsic pathway can activate an enzyme called caspase 8. Your extrinsic pathway activate an enzyme called caspase 9 through a complex called apoptosome. We'll discuss that later. So both of these are initiator caspases. And then your initiator caspases can then activate what is called executioner caspases. So you have executioner caspases, and these executioner caspases then, or they're also called effector caspases, they can cleave substrates. These substrates are structural components of the or different signaling proteins, DNA, for example, or they don't directly cut the DNA, but they cause the cleavage of the DNA. So this actually causes apoptosis. So we'll discuss extrinsic as well as intrinsic pathway in detail, and we'll discuss the component, especially the BCL2 family members that are involved in apoptosis. So this is just one example. It can be BAX, or it can be BAC, or several other proteins. So this is how an apoptotic cell looks like. So this is a normal viable cell. This is phase contrast image. And this is when you treat the cell with the daunorubicin, a DNA damaging drug at 10 micromolar for 12 hours. So you see the cell has shrunk. And there's a movie for that as well. So if you see, so these cells are undergoing apoptosis. So you will see that these cells will shrink in a while. And this is, and the small blebbings like these, and these are kind of what I call apoptotic bodies. So it's like, the cell is kind of boiling. So you can see that the nucleus is also fragmented. So the DNA is fragmented, nucleus is fragmented. And also you see that the mitochondria, they release cytochrome C. So here you see cytochrome C in the mitochondria, and here you see that the cytochrome C has been released. So we'll talk about that later. So very important component of apoptosis are the caspases. So these are specialized specialized proteases. They're called caspases because they are cysteine dependent aspartate specific proteases. So cysteine dependent, so their catalytic site has a catalytic cysteine, and they cleave at the aspartic acid site, and they are proteases. So that's why they are called caspases. The cysteine actually residue acts as a nucleophile and cleaves the substrate after the aspartate. So there are 14 mammalian caspases. So there are 14 caspases. 11 of them are present in humans. There are two types of caspases, apoptotic caspases and inflammatory caspases. The apoptotic caspases further can be divided into two categories. So they can be initiator caspases. We talked about initiator caspases, and we said that initiator caspases are caspase 2, 8, and 2, 8, 9, and 10. While the other caspases called effector caspases, they are caspase 3, 6, and 7. These are apoptotic caspases. Now the inflammatory caspases, they are caspase 1, caspase 4, caspase 5, caspase 11, 12, 13, and 14. They play roles in inflammation. So they can, some of them, they can be part of the what is called inflammasome as well. So the initiator caspases, they have an additional N-terminal part. So this is, if this is the caspases, they have an additional N-terminal part about 90 amino acid long, and this can be, this domain can be death effector domain, or this can be caspase recruitment domain. So they have a N-terminal card domain or death effector domain. So we'll talk about that. So all the caspases, they are synthesized as inactive proenzyme, and these proenzymes are activated into active caspases through various processes in various signaling complex. We'll talk about that later as well. So based on their substrate specificity, they can be classified into groups. So group 1, group 2, and group 3. So they recognize amino acids that are N-terminal to the aspartate, and they cleave them accordingly. So I have shown here कि कौन सी caspases कौन से amino acids पे cleavage cause करती है. Okay, so as I mentioned, there are two caspases, two types of caspases, effector caspases, shown here by the example of caspase 3, and initiator caspases shown by example of caspase 8 and caspase 9. As I mentioned, the initiator caspases, they have an N-terminal additional part that can be death effector domain, or that can be caspase recruitment domain or CARD domain. The effector caspases, they have also kind of a pro domain here, which can be cleaved, which is actually cleaved when the caspase becomes active. So this red part, it shows cysteine, which is required for the nucleophilic attack on the aspartate. And then you have these loop 1, loop 2, loop 3, loop 4. They form the active site. And you can see that cysteine is present at the start of the loop 1. Now, cleavage of the caspase. So these sites, the arrows, they show the cleavage site. So you can see that what you end up with a smaller fragment here, which can be, which is the beta part. So this is the beta part, and this is the alpha part, which is the larger fragment. So what happens is when the caspase is cleaved, so this, for example, the pro domain is cleaved, and then you have a cleavage here, actually two cleavages here as well. That releases a large subunit, which is called alpha chain, and then a smaller subunit, which is called beta subunit or beta chain. So these alpha and beta then heterodimerize. So you get this. So these alpha and beta, they heterodimerize, and this is your active site that contains the cysteine. And then these heterodimers, they from two different caspases, they bind together to form an active caspase dimer, this which is technically a tetramer. So the active caspase is always a dimer of the two caspases. Each monomer of the dimer contains the alpha and the beta chain. Excuse me. Okay. So how the caspases are activated and how is that activation regulated? So caspases, the activation of caspases, because they can cleave very important cellular structures, their activation is highly regulated. So it's very strictly controlled. So there are actually two levels of activation or regulation of caspase activation. One is conversion of caspase precursor, that is the cleavage of the caspase itself to convert it into active form. And the other one is through inhibition by natural inhibitors. So there are inhibitory proteins such as IAP proteins, which is inhibitor of apoptosis. So these proteins, they are direct inhibitors of caspases. So they can actually inhibit caspases as well. So the first one, the conversion of caspase precursors into active form, is mediated in large protein complexes called death platforms. And there are three death platforms. One is called pidosome. So this activates caspase 2, which is, as we learned, is an initiator caspase. The apoptosome activates caspase 9, and the DIS complex, which is death inducing signaling complex, activates caspases 8 and 10. Now, these death platforms, they only activate initiator caspases. Now, your pidosome, we will not talk about this. This says, P53 induced protein. So it's composed of two proteins, P53 induced protein with death domain. Another protein is called RAIDD, which is RIP associated ICL homologous protein with death domain. So, you don't need to remember that. We're not studying pidosome in detail. What we are studying in detail are these two death platforms called apoptosome and caspase 9. So your apoptosome, as you can see, activates caspase 9 in response to intrinsic pathway. Is part of the intrinsic pathway. So your apoptosome is intrinsic pathway. Your disc complex, death-inducing signaling complex, is activated in response to extrinsic pathway, is part of the extrinsic pathway. So once the caspases are activated, what are their substrates? So caspases can cleave other caspases. So other pro-caspases, for example, your initiator caspases will cleave the effector caspases or executioner caspases. It can be pro and anti-apoptotic protein. For example, caspases can cleave BCL2 family of proteins, and their C terminals can. So BCL2 family of proteins are usually anti-apoptotic, but once they're cleaved, their C terminals can act as pro-apoptotic. Similarly, caspases can cause, activation of ICAD. So it's inhibitor of, they can cleave ICAD that causes the activation of DNases. So ICAD is cleaved by caspases. So ICAD is inhibitor of caspase activated DNases. So this protein, ICAD, inhibits DNases. And when caspases are activated, they they cause cleavage of ICAD. So the DNases are activated. So these DNases then cause fragmentation of the DNA or cleavage of the DNA. Your caspases can also cleave structural protein. For example, they can cleave gelsolin as well as laminin, which is present in the nucleus. They can also cleave important signaling protein. For example, proteins like PKC delta. They can cleave kinase like V1, which is involved in DNA damage response. They can also cleave RB, which is a protein involved in cell cycle. They can also cleave PARP, poly-ADP ribose polymerase, which is involved in DNA repair. So now we talk about BCL2 family of proteins. So BCL2 family proteins are key proteins that regulate apoptosis. They can be both pro-apoptotic or they can be anti-apoptotic. And actually there is a threshold between, or the ratio between anti-apoptotic and pro-apoptotic proteins that actually regulates apoptosis. So one family, so they can be divided into three kind of subfamilies. One of them is are the pro-survival proteins. So they are anti-apoptotic proteins. And this includes BCL2, BCLXL, BCLW, MCL1, A1, and BCLB. So these proteins have BCL homology domains. So 4 of them, BH1, BH2, BH3, and BH4. And they have a C-terminal hydrophobic transmembrane domain. Apart from that, they are also these alpha helices that we will talk about later. And then you have pro-apoptotic proteins called effectors. They includes BAX and BAC, which are two most important, and a third member called BOC. So they also have a transmembrane domain and BH1, BH2, and BH3 domains. Some papers write they also have a BH4 domain, but most of these proteins, they only have BH1, BH2, and BH3 domains. The last one, or the most abundant family, is the BH3 only family. As the name suggests, they have only BH3 domain. They don't have BH1, BH2, or BH4 domains. This includes BIM, PUMA, BAD that we studied when we studied the AKT pathway, AKT phosphorylates, BAD, remember that? NOXA, BIC, and BID. So BID is very important. We'll talk about that later. So the the BCL2 family proteins, they form a BCL2 structural fold in which a hydrophobic alpha helix, the alpha helix 5, this one is surrounded by 7 different amphiphilic alpha helices. So in the center you have this hydrophobic alpha helix 5, and on the sides you have 7 different amphiphilic alpha helices. Only 5 of them are shown in this figure, and this makes a surface groove. So this is for the BCLXL. So BCL2 has a similar surface. So when the BH3 only proteins bind, for example, here you can see that the BIM, this shown here. So BIM's BH3 domain, it can bind on this surface groove. And this binding involves 4 hydrophobic residues shown here: hydrophobic 1, hydrophobic 2, hydrophobic 3, and hydrophobic 4. And it also involves a salt bridge between the negatively charged aspartate and a positively charged arginine on the surface. Now, we know that the BH3 only proteins, they inhibit the BCL pro-survival protein. Like, for example, we talked about that BAD binds to BCL2, and this causes inhibition of BCL2. So BAD is inhibiting BCL2, and when AKT phosphorylates BAD, the BAD is bound to 1433, and BCL2 is then released, and this is how AKT inhibits apoptosis and AKT pathways pro-survival. So this is another slide that shows you the same. So you have anti-apoptotic BCL2 family members: BCLXL, BCLW, MCL1, A1, and BCLB. They have BH1 domain, BH2, BH3, and BH4, and they have a transmembrane domain. They bind to the cytosolic parts of the membranes, especially the mitochondrial membrane. Okay, and then you have pro-apoptotic proteins like BAX and BAC. They form actually a channel, apoptosis inducing channel in the membrane of the mitochondria, the outer membrane of the mitochondria, and they also have a BH1 domain, BH2, and BH3 domain, but they lack this fourth BH4 domain. They also have a C-terminal transmembrane domain that contains hydrophobic alpha helix. The BH3 only proteins, they, as I mentioned before, they all have a single BCL2 homology domain, that is BH3, and some of these, they also have a transmembrane domain, but rest of them, they don't have any kind of transmembrane domain. So why there are so many of the BCL2 family? Because of their specificity. So for example, BIM, PUMA, and TBID, they can inhibit all the pro-survival BCL members. BAD can inhibit only these 3, and NOXA can only inhibit these 2. Similarly, your pro-survival protein, pro-survival BCL2 members, pro-survival BCL2 members, okay, they inhibit back and back. So these 5, they inhibit only backs, and these 3, they only inhibit back. So, there's some specificity. Similarly, these are the BH3 only members. So PUMA and NOXA, we know they can be activated by P53. So, this slide actually shows you different stimuli that can activate apoptosis by increasing the concentrations of these BH3 only proteins. For example, any DNA damage will increase the concentration of P53, and P53 is a transcription target, and PUMA and NOXA are the transcriptional targets, is a transcription factor. P53 is a transcription factor, and PUMA and NOXA are the transcriptional targets of P53. And all of these, they inhibit the BCL2 family of pro-survival proteins. So what is the function of BH3 only protein? They cause neutralization of the pro-survival BCL2 family protein. We talked about this. We said that bad, abhi maine piche example di, bad binds to BCL2 and this inhibits BCL2. So when bad is phosphorylated by AKT, this binds to 1433 and this releases BCL2, which inhibits apoptosis by inhibiting back and backs. So it inhibits back and backs, right? So it neutralization of the pro-survival proteins. The second is direct activation of the pro-apoptotic protein. So we said BH3 only proteins, what do? They inhibit, they inhibit BCL2, and BCL2 ये क्या कर रहा है? It is inhibiting backs and back, which are causing apoptosis. So back and backs are pro-apoptotic, so they're causing apoptosis, and BCL2 is inhibiting back and back, so it is inhibiting apoptosis. BH3 only is inhibiting BCL2, so it is causing apoptosis. Now, BCL2 only can also directly activate back and backs, and this can actually cause apoptosis as well. So this can increase apoptosis as well. Now, let's talk about the mitochondrial apoptosis pathway. So you have, let's say you have increase in the concentration of BAD. So BAD can be increased by excessive growth factor. Oh, sorry, it can be increased by growth factor deprivation. So BAD is a BH3 only protein. Similarly, your DNA damage can increase the concentration of backs. So what they do, BAD and backs, we just found out that they increase. So if you have mitochondria, so this is outer mitochondrial outer membrane, and then you have mitochondrial inner membrane, right? So here, you have these back, Excuse me, so I'll have to redraw that. Okay. So here you have back, back, and backs. They form the mitochondrial apoptosis induced channel, or MAC. It is called mitochondrial apoptosis induced channel, MAC. So BAD and BACs, they facilitate this channel. So let's say there are many channels throughout the mitochondria. So what happens is that there is this protein called cytochrome C. So you get release of cytochrome C. So this protein is cytochrome C. So this cytochrome C binds to a protein called APAF1. So let's say this is the protein called actually, let me draw it a little bit on the side. So APAF1. So actually it binds to 7 different molecules of APAF1. 1, 2, 3, 4, 5, 6, and 7. And then 7 different molecules of let's say cytochrome C bind here. And then it also binds to 7 different molecules of caspase 9. So you have caspase 9, caspase 9, caspase 9, caspase 9, caspase 9, 9, caspase 9. So this is a PAF1, this is caspase 9, and this is cytochrome C. So this complex, which has 7 proteins of each, is called apoptosome. So what your apoptosome does is causes the cleavage of caspase 9. So your caspase 9 actually becomes active. So your active caspase 9 then causes the, so you have pro-caspase 3, 6, and 7, which are effector caspases. So caspase 9 cleaves these procaspases into active caspases. So active caspase 3, 6, or 7. And this can actually cause apoptosis by cleaving various substrates. So it cleaves substrates. Now, in addition to cytochrome C, many other proteins are released. For example, a protein called diablo or SMAC are released. These proteins actually are, they bind to a protein called inhibitor of apoptosis. So there's a protein called IAP, inhibitor of apoptosis. This actually directly inhibits apoptosis. So these diablosmacs, they inhibit IAPs. So you see on one side, cytochrome C is released, caspase 9 is activated. On the other side, the mitochondria also release other proteins that make sure that caspase 9 is not inhibited by IAPs. So inhibitor of apoptosis. Similarly, other enzymes, other proteins that are released, for example, endonuclease G is released. So this also cleaves DNA, causes cleavage of the DNA. Another protein that is released is apoptosis inducing factor, so AIF, that causes so this is also released from the MAC. This causes chromosomal condensation and cleavage of DNA. So this is how your intrinsic pathway works. So you have growth factor deprivation. You have DNA damage that induces BH3 only proteins. I will come to death receptors later. And these proteins, BH3 only proteins, they cause increase in this MAC, mitochondrial apoptosis induced channel. So what does this MAC does is that it releases cytochrome C, which binds to APAF1, caspase 9, 7 molecules of each, and this causes the activation of caspase 9. So from pro-caspase 9, it is converted into active caspase 9. So active caspase 9 can be inhibited by IAPs. So IAPs, as I mentioned, they are inhibited by diablo-SMAC. So caspase 9 converts pro-caspases, the जो effector caspases है, into active caspases. So this is how your intrinsic apoptotic pathway works. That is also called mitochondrial apoptosis. Okay, so another very important thing here is that the APAF contains a card domain, so caspase recruitment domain, and this card domain makes a homotypic interaction and this card domain is a homotypic interaction with the card domain of caspase 9. So the card we studied in one of the previous slides that the caspase 9 has a N-terminal card domain, and caspase 8 and 10 have an N-terminal death effector domain. So the card domain of caspase 9 binds to the card domain of APAF1, and this homotypic interaction is required for this apoptosome formation. Now we come to the extrinsic pathway. If I draw a membrane here, so we studied about the TNF receptors, and we said, they have the death domains. Similarly, the FAS receptors we also studied. They have, extracellular card domain, and they always act in kind of trimers. So they have these intracellular domains, and then they have these intracellular death domain. So if I draw this death domain as yellow here, so this is death domain, and this is your FAS receptor, and then the FAS ligand also binds in trimeric form. So your receptor is in trimeric form, and your ligand is in trimeric form. Now, once the ligand binds to the receptor, the receptor recruits another protein called FAD. So FAD is for FAS associated death domain. Let me correct this. So FAD is FAS associated death domain. So it has a death domain, and then it has another domain called death effector domain. So your FAD has a death effector. So there are three. I'm just drawing two of them for convenience. So this is death effector domain. So now you see this death effector domain of FAD binds to the death effector domain of FAS receptor, and this is homotypic interaction, as I just described. Now, on previous slide, I described that caspase 8 and caspase 10, they have a death effector domain at their N-terminus. And this death effector domains then makes homotypic interaction with the death effector domain of FAD. So you get, and this is the caspase part, the catalytic part that is also cleaved into two parts. So, you have this homotypic interactions, and this complex as a whole is called death inducing signaling complex that we talked about. So this is a death platform. Now, once this death platform is formed, your caspase 8 becomes active. ट्राइमेरिक फॉर्म में वही जो टेट्रामेरिक फॉर्म में वही जो हमने बात की थी कि ये एक्टिव हो जाती है, एंड दिस कैन देन क्लीव जो आपकी कैस्पेज प्रो कैस्पेज पेज इफेक्टर कैस्पेजेस हैं 3, 6 और 7, उनको कन्वर्ट कर देती है एक्टिव कैस्पेजेस में, एंड दीज एक्टिव कैस्पेजेस देन क्लीव substrates, and this can cause apoptosis. Now, one of the substrates of this active caspase 8 is a protein called BID. So BID जो है वो क्लीव होके truncated BID बन जाती है. So truncated BID जो है उसके अंदर उसका एक पार्ट क्लीव हो जाता है, and this truncated BID then can activate the mitochondrial pathway. So it can activate the mitochondrial pathway. So now if we go back to the previous slide here, so you see I didn't explain this. So ये जो आपका death receptors हैं, they can cause activation of caspase 8, and your caspase 8 can cleave BID into T-BID, and this T-BID can then activate the MAC. So it can cause the activation of or release of the cytochrome C, which can activate the apoptosome, and your caspase 9 is activated, so your mitochondrial pathway is activated. So the take home from this is that your extrinsic pathway can activate your mitochondrial or the intrinsic pathway. So now this is to summarize the extrinsic pathway. So you have a fast ligand that binds to the fast receptor that has a death domain, and these death domain, they make homotypic interactions with the death domain of the FAD, and the death effector domain of FAD makes homotypic interaction with the death effector domain of your caspase 8. Caspase 8 then becomes active caspase 8, and this can cleave pro-caspase 3 into active caspase 3. It can also cleave BID into truncated BID, which can induce mitochondrial apoptosis, and your active caspase then can induce apoptosis. Okay, so that's it for today's lecture, and I will be online on Wednesday at 5 o'clock, and I will send you the link for the Zoom meeting. Thank you very much.